Welcome to the Deep Dive, where we plunge into complex topics armed with the best sources to uncover surprising facts and distill truly useful insights. Today we're tackling one of humanity's grandest ambitions, extending healthy life. This isn't just a biological puzzle, It's a quest with profound scientific, medical, philosophical, and even economic implications. We're going to unpack the fascinating journey of geroscience, the study of aging, from laboratory breakthroughs to the challenging reality of human clinical trials. Our guide for this deep dive is Aging Therapies, Human Trials, Endpoints, and Regulatory Realities, a really rich collection of insights from leading experts who are, quite literally, shaping the future of healthy longevity. So what's our mission today? We're going to explore the critical disconnect between what works beautifully in the lab and what actually translates to human benefit. This leads to what some call beautiful failures. We'll then examine how regulators currently view aging and how that stance dictates the entire drug development strategy with a look at trials like TAME. We'll clarify the often confused terms biomarker and surrogate endpoint and understand why that distinction is, vital. Then we'll dive into the ingenious ways researchers are designing trials to measure genuine health benefits in humans, followed by the crucial role advanced biomarker panels play. After that, we'll shift to the practicalities of trial design, from who gets enrolled to innovative adaptive approaches. Safety, especially for drugs given to largely healthy older adults, is paramount, so we'll definitely cover those unique challenges. We'll also touch on the often overlooked manufacturing hurdles, particularly for cutting-edge cell and gene therapies. Finally, we'll consider the broader policy and economic landscape, asking how we ensure health span extension becomes a public good, accessible to all. This isn't just a quick summary. It's a deep intellectual exploration into the nuts and bolts of how we might one day unlock healthy longevity for everyone. Okay, here's where it gets really interesting. We've seen these incredible longevity benefits in lab mice with dozens of different interventions. So why isn't this already happening for us? It feels like we should be taking a pill by now, right? Given all those successes.
It's a fantastic question, and it really gets to the core paradox in geroscience. The apparent ease of extending mouse lifespans stems from a few key factors. First, mice have naturally short lifespans, typically, 2 to 3 years. This makes it pretty straightforward to observe the full impact of an intervention on their overall longevity within a reasonable experimental timeframe. You can wrap up a mouse study and potentially see significant lifespan extension in just a year or two. Second, these mice live in highly standardized, controlled environments. They're free from many of the confounding factors that influence human health, like varied diets, different lifestyle choices, environmental exposures, pre-existing conditions, you name it. This allows for a very clean observation of a drug's direct biological effect. But now, contrast that with humans. We're talking about a typical lifespan of 80 years or more. Waiting decades to measure a treatment's effect on overall human lifespan, or even just the onset of specific age-related diseases, is simply impractical. It's financially prohibitive and ethically challenging for clinical trials. This fundamental difference leads to what researchers, quite poignantly, call beautiful failures.
Beautiful failures. That's quite a term. What exactly does it mean?
It really encapsulates the paradox. Many once promising longevity drugs, despite showing elegant scientific success in animal models, have simply failed to translate into tangible, patient-relevant benefits in human trials. The core reason. It's that the mechanism of benefit that was observed and celebrated in mice just didn't translate into meaningful improvements in how human patients feel, function, or survive. So a drug might extend mouse lifespan by affecting a particular pathway, let's say, but that pathway's impact might be different or less pronounced, or maybe it's just outweighed by other factors in a complex human system. It really highlights that for a human therapy to be considered successful, it absolutely must genuinely improve the patient's lived experience, not just tweak a biological marker in a lab animal.
Right. So it's not just a simple matter of scaling up the dose from a mouse to a human. This requires a complete reevaluation then.
Absolutely. It's a whole paradigm shift. This challenge has necessitated a fundamental shift in how we design human trials. We're essentially redesigning these trials from the ground up to avoid these beautiful failures. This means rethinking primary endpoints, what we actually measure to determine success, and critically examining the role of biomarkers as indicators of biological changes. There's also a complete reassessment of the broader regulatory strategy for identifying genuinely effective geroprotective therapies. The ultimate goal is to find ways to identify truly effective therapies for humans, despite our long lifespans and the immense complexities of human biology and our environment. It's really about designing trials that are both scientifically rigorous and practically feasible.
That makes perfect sense. So if we can't just apply mouse models directly, and the
Sure.
goal is really to improve how we feel, function, or survive, what does this mean for how regulators view aging itself? It sounds like that's the very first sort of foundational hurdle.
That's precisely right. This is perhaps the most fundamental regulatory hurdle in the entire field. Regulatory bodies like the FDA in the US or the EMA in Europe currently do not recognize aging itself as a defined treatable disease or a standalone medical indication. Their model, the way they approve drugs, is structured so drugs are approved to treat specific, well-defined diseases, things like cancer, diabetes, heart disease. Aging, instead, is understood as a major risk factor for numerous diseases, but not a standalone target for drug approval. And this distinction, it's absolutely crucial because it dictates the entire framework for how a potential anti-aging therapy must be developed and presented for clinical trials and eventual market approval.
Okay, so a company can't just walk in and say, our drug treats aging. They have to be more strategic about it.
Exactly. Any therapy aiming to extend health span, those years lived in good health, must be developed and framed under a different, already accepted banner. This is a crucial strategic pivot developers have to make. Instead of claiming to treat aging, trials must strategically aim to reduce the risk of specific age-related conditions, or maybe improve a measurable functional outcome. Their goals have to be expressed in terms of prevention or treatment of specific age-related diseases or syndromes, very carefully avoiding claims of directly treating aging. This strategic framing shapes the entire trial design, really, from who gets enrolled to what endpoints they choose.
Can you give us a concrete example of this strategic thinking in action, something that shows how this works practically?
Yeah, absolutely. The TAME trial targeting aging with metformin is the prime example here. Rather than seeking approval for metformin as an anti-aging drug, TAME is meticulously designed to demonstrate that metformin, which is a widely used diabetes drug, can delay the onset of several major age-related diseases simultaneously. Its design specifically targets the prevention of multimorbidity in older adults, and it lists precise components it's trying to prevent: myocardial infarction, stroke, cancer, heart failure, dementia, or mild cognitive impairment or death. These are all well-recognized serious age-related conditions. And crucially, the FDA reviewed and approved the design of TAME on this precise basis. That validated this innovative approach of targeting the prevention of multimorbidity in older adults. That approval was a really landmark moment, providing a kind of template for navigating the current regulatory landscape.
So if TAME succeeds, that could potentially open the door for a lot of other interventions targeting aging mechanisms.
Potentially, yes, that's the hope. If TAME yields positive results, it could serve as a powerful proof of concept. It could prove that aging, as represented by multimorbidity or frailty, can serve as a legitimate therapeutic indication. And that success could then pave the way for regulators to maybe broaden their perspective, potentially considering multimorbidity or frailty as approvable drug targets in the future. But until such a shift occurs, companies have to make shrewd strategic choices. For example, a senolytic drug one designed to clear senescent or zombie cells might initially seek approval for specific, already recognized diseases of aging, things like idiopathic pulmonary fibrosis, a nasty lung disease, or maybe osteoporosis. By first gaining approval for a specific disease, companies can build a strong evidence base before trying to tackle broader healthy aging outcomes.
Right. It sounds like a clever workaround to the current system, but still firmly rooted in showing a tangible benefit that matters to patients.
Exactly. And regardless of the specific framing, the FDA consistently reinforces its explicit requirement. Any approved drug must improve how patients feel, function, or survive. This core principle directly shapes the selection of primary endpoints and the use of biomarkers in trials. It mandates that the benefit must be tangible and clinically meaningful from the patient's perspective. It's a pragmatic approach, really, to a very complex biological process.
Okay, so if the goal is to make us feel, function, and survive better, and we can't directly target aging, how do we actually measure that a drug is working? And how do we do it efficiently, without waiting decades? This brings us to that subtle but crucial distinction between a biomarker and a surrogate endpoint. What's the difference?
You're right. The distinction is subtle, but it's absolutely profound when it comes to drug approval. Let's start with biomarkers. Think of a biomarker as basically any objectively measured indicator of a biological process or state. It's like a snapshot of what's happening inside your body at a specific moment. This could be something simple like your cholesterol level from a blood test, or something more complex like a specific protein in your blood or an imaging result. Now, the FDA has a formal biomarker qualification program, or BQP. This program essentially gives a biomarker a sort of seal of approval for use in a specific context within trials. For instance, a plasma protein level might be qualified to help select patients for a trial, maybe only enrolling those with high inflammation, or maybe as a secondary outcome measure to track alongside the main clinical goal.
Okay, so it's a useful tool for the research, but it's not proof in itself that a drug is working.
Precisely. A qualified biomarker is primarily a development tool. It's useful, it's validated for that specific use, but it does not imply that merely changing this biomarker is sufficient for drug approval. It's useful for research, for understanding mechanisms, for maybe enriching a trial population, but it doesn't stand alone as proof of efficacy for approval. Now, a surrogate endpoint is different. It's a specific type of biomarker or maybe an intermediate clinical outcome that is used in place of a true clinical outcome in a trial. The critical assumption here, and it's a big one, is that changes in the surrogate reliably predict real clinical benefit. Their main purpose is to significantly speed up trials, especially when waiting for the true clinical outcomes, like overall survival, progression to disability, or maybe developing a rare disease would simply take too long.
Like blood pressure for predicting stroke risk, or maybe HbA1c levels for diabetes complications. Those are classics, right?
Exactly. Those are perfect examples. Blood pressure reduction is widely accepted as a surrogate endpoint for stroke prevention because numerous large trials have definitively proven that lowering blood pressure significantly reduces stroke risk. Similarly, HbA1c level is used as a surrogate for microvascular complications in diabetes, and the FDA has acknowledged their importance. Something like 45% of new drug approvals between 2010 and 2012 were based on surrogate endpoints, especially in areas like cancer and heart disease.
So the core difference is really what they can be used for in drug approval. A biomarker can help research, but a surrogate endpoint can actually stand in for the final clinical outcome for approval.
That's the absolute key distinction. Qualification is about a biomarker's general usability as a reliable research tool in a specific context. Acceptance as a surrogate, however, is about using that biomarker as the primary basis for granting drug approval. The bar for becoming a surrogate is much, much higher. It must be validated by compelling evidence showing that improvements in the biomarker predict a real, tangible clinical benefit, how the patient actually feels, functions, or survives. Regulators accept surrogates when they are fully validated, like blood pressure, or sometimes under accelerated approval if they are deemed reasonably likely to predict benefit. Though, in those accelerated cases, confirmatory trials are always required post-approval to nail down that clinical benefit.
Okay, that's clear. And for aging specifically, where do we stand with these? Do we have any validated surrogates yet?
That's the million-dollar question. For aging research, we might, for example, get an epigenetic aging clock, which measures biological age from DNA patterns qualified by the FDA as a risk predictor. This would mean researchers could reliably use it in trials to identify individuals aging faster or to track biological changes in response to an intervention. It becomes a validated tool. However, and this is crucial, no aging biomarker is currently validated as a surrogate endpoint. This means the FDA would not approve a geroprotective drug simply because it showed, say, a slowing of an epigenetic clock without robust accompanying clinical outcome evidence. So for now, biomarkers like epigenetic clocks play vital supportive roles in aging trials. They can provide proof of a drug's mechanism of action, help predict long-term risk, or help stratify patient populations for enrollment. But the primary endpoints for approval must be clinical outcomes that directly measure how patients feel, function, or survive. Now, the field is actively working incredibly hard to validate true surrogate markers of aging, but experts caution that it's a long jump from merely showing a biomarker correlates with age to definitively proving that changing that biomarker alters meaningful clinical outcomes. It's a high bar.
Right, makes sense. So if we can't rely on just a biomarker for approval, and we can't wait decades for lifespan data, how are researchers even defining success in these human aging trials? What are they actually measuring to show a drug is working within a reasonable time frame? This is where the ingenuity really comes into play, I guess. #tag4, defining meaningful endpoints for aging trials.
It really is a fascinating area innovation right now because aging isn't a single disease and tracking individual age-related diseases one by one is just too inefficient for a trial. Researchers need endpoints that can capture overall aging benefits within a practical time frame. So the field is increasingly focusing on clever approaches like composite endpoints and functional outcomes. These aim to reflect holistic health span improvements, recognizing that aging is really a multi-system decline. It's about finding ways to measure the prevention of that whole cascade of vulnerabilities that comes with age, not just one isolated condition.
Okay, so give us some examples of these creative endpoints. What are people actually using in trials?
Certainly. One key approach that's gaining a lot of traction is time to multimorbidity. Instead of just tracking, say, the first heart attack, this endpoint measures the time until a participant develops any one of a predefined set of major age-related diseases or dies. The TAME trial we mentioned is the perfect example. Its primary endpoint is exactly this: the time to the first occurrence of any of 6 specific events: heart attack, stroke, cancer, heart failure, dementia, or mild cognitive impairment or death.
And what's the big advantage of using a composite approach like that?
Well, there are 2 main advantages. First, it greatly increases the number of events you observe in the trial compared to tracking just one disease. This significantly improves statistical power and means you can potentially run a shorter trial with fewer people. Second, it directly aligns with the core geroscience hypothesis. A therapy that truly targets fundamental aging mechanisms should, logically, delay multiple age-related diseases simultaneously, not just one. So the sheer breadth of the outcome makes it highly relevant and compelling, both for patients who want more healthy years and for policymakers looking at the overall disease burden. Another really important category is the onset of mobility or functional disability. This focuses squarely on maintaining physical independence and functional capacity, things that really matter to older adults. Outcomes might be something like major mobility disability, often defined as being unable to walk 400 meters, or perhaps disability-free survival, which means being alive without persistent physical disability or dementia. These endpoints resonate very strongly with older adults' real-life goals: staying active, independent, and self-sufficient. While defining and measuring disability can sometimes be tricky, their direct patient relevance makes them very powerful endpoints.
What about broader health indicators? Things that reflect overall well-being beyond just specific diseases or functions.
That leads us to another interesting option: hospitalization-free survival. This measures the time someone lives without being hospitalized for any reason, effectively aggregating major health events that require acute care plus death into a single metric. It implicitly captures the worsening of various underlying morbidities because many conditions, when they become severe, lead to hospital stays. It's quite compelling because, let's face it, older adults really prioritize staying out of the hospital. And it also reflects healthcare utilization, which is obviously of great interest to payers. It's a clear indicator of overall morbidity compression. And finally, researchers are exploring the frailty index or composite frailty outcome. Frailty is understood as a state of increased vulnerability due to the accumulation of deficits across multiple physiological systems. A frailty index sums up a long list of health variables, symptoms, signs, lab abnormalities, functional measures, into a single continuous score. Trials could then use the change in a validated frailty index over time as an endpoint. The beauty of the frailty index is that it's a continuous measure, potentially sensitive to even small health declines, and it inherently combines multiple body systems, which aligns perfectly with the geroscience approach of targeting multisystemic aging. Now, there are challenges with standardization. Different indices exist, but success on a well-constructed frailty endpoint would powerfully demonstrate improved resilience and overall health in older adults.
Okay, so it sounds like by combining these different types of outcomes, researchers are trying to get the best of both worlds, statistical power, and a really comprehensive picture of whether a drug is genuinely improving health span.
Exactly. The choice of endpoints is absolutely crucial and must be thoughtfully aligned with the drug's expected mechanism of action. Many geroscience trials now use a primary composite endpoint, like time to multimorbidity, but they also wisely track the individual components separately as secondary outcomes. This helps understand which parts of the composite are being driven by the treatment. They also often include key secondary endpoints focusing on specific domains like cognitive function, walking speed, or maybe immune function to capture more nuanced benefits. Ultimately, the chosen endpoints must be persuasive to all key stakeholders, patients, regulators, and payers, by demonstrating tangible benefits, reduced hospitalizations, improved independence, clear indicators that align with that core mandate of feel, function, or survive.
This is brilliant, really thinking about aging not as one single thing, but as this cascade of vulnerabilities, and then designing ways to actually measure the prevention of that cascade. It's very clever. But how do we know if a drug is actually hitting the underlying biological aging process? This sounds like where biomarkers come back into play. Maybe not for approval, but in a very specific supportive role.
You're absolutely right to highlight their continued importance. They play a critical supporting role. While the primary endpoints must be clinical, reflecting that feel, function, survive mandate, most geroscience trials now include a wide array of biomarkers of aging as secondary or exploratory outcomes. These biomarkers serve two critical functions in these trials. First, demonstrating proof of mechanism, basically, showing that the intervention actually hit its intended biological target within the body. Did it do what we thought it would do biologically? And second, offering early signals or predictors of a drug's potential long-term benefits, even if these biomarkers aren't yet validated surrogates for approval themselves. And there's sort of an emerging consensus suggesting including specific categories in a standard panel: epigenetic clock measures, proteomic signatures, and inflammatory markers.
Okay, let's break those down. Start with epigenetic clocks. We touched on them, but what exactly are they measuring?
Right. DNA methylation clocks, or epigenetic aging clocks, are these really sophisticated tools that measure biological age based on specific patterns of DNA methylation. These are chemical tags on your DNA that change predictably with age. A great example that's getting a lot of attention is denidin-pace. What's neat about denidin-pace is that it measures the rate or pace of aging, not just your static biological age at one point in time. It gives a single number readout of how fast an individual's biological aging process seems to be occurring right now. And it's been shown to correlate strongly with things like multimorbidity, physical decline, and even mortality risk. So in a clinical trial, seeing a slowing of denidin-pace in the treatment group compared to placebo would provide pretty compelling evidence that the therapy is truly decelerating the underlying biological aging process. Including at least one DNA methylation clock like Dundon-Pace or others like Grimmage is becoming pretty standard practice in these trials. But again, just to reiterate, no clock is yet a validated surrogate endpoint for approval. They provide powerful supportive evidence.
Okay, that makes sense. And what about looking at the body's proteins? You mentioned proteomic signatures.
Yes, that's our second key category: proteomic and metabolic signatures. The aging process leaves distinct fingerprints in the blood proteome, which is just the full set of proteins circulating in your blood at any given time. Levels of various inflammatory proteins, metabolic proteins, growth factors, they all tend to change in predictable ways with age. Things like GDF15, IL-6, TNF-alpha, IGF-1, these are often mentioned. Modern high-throughput proteomic platforms can now measure hundreds or even thousands of these proteins at once, generating a comprehensive aging signature. So trials often include at least one proteomic panel to assess if a therapy can reverse an aging-related protein profile. For instance, you might expect a drug mimicking caloric restriction to lower chronic inflammatory markers. And the expert consensus here is really that no single biomarker will suffice. You really need a panel of multiple biomarkers capturing different hallmarks of aging to get a robust and comprehensive picture of an intervention's effect. Some researchers talk about a basket of biomarkers.
And I imagine inflammation itself is a big target here, given the concept of inflammaging.
Absolutely. That brings us to our third category: inflammatory and immune markers. Chronic low-grade inflammation, or inflammaging, is recognized as a key hallmark of aging, and it's implicated in many age-related diseases. So trials routinely track common inflammatory markers like C-reactive protein, CRP, IL-6, TNF-alpha. A geroprotector with known anti-inflammatory action would ideally show significant reductions in these. And for therapies that are designed to modulate the immune system more directly, monitoring effects on immune cell populations like T-cell subsets, or perhaps more functionally, looking at the response to vaccination becomes crucial. We mentioned earlier that some mTOR inhibitor trials have actually measured changes in older adults' immune response to the flu vaccine as a functional immune biomarker. Including an immune panel helps capture these key aspects of aging biology and provides important mechanistic support if improvements are seen. It also serves as a safety check.
Okay, so these biomarkers are incredibly valuable for understanding how a drug works, whether it's hitting its target and its potential long-term impact, even if they aren't the primary measure for getting the drug approved.
Precisely. They cannot by themselves prove efficacy for drug approval that still rests on the clinical endpoints, but they enrich the trial data immensely. Think about it. If a clinical endpoint isn't met, maybe due to trial length or size, but the biomarkers show significant positive changes, like slower epigenetic aging, lower inflammation, that could provide a very strong rationale to justify a larger, longer, or maybe redesigned follow-up trial. Biomarkers are also essential tools during development for dose selection and target engagement, basically confirming if a chosen drug dose is actually shifting the aging biology in humans as expected. And they're increasingly powerful for stratification for precision geromedicine, helping to identify specific subgroups of patients, maybe based on their baseline biological age or inflammatory status, who might benefit most from a particular intervention. So the overall trend is definitely towards using a multimodal biomarker panel, combining epigenetic, proteomic, inflammatory, and maybe other markers to really help triangulate whether an intervention is truly affecting the biological aging process during the course of the trial.
Okay, that paints a really clear picture. We know what we need to measure both clinically and biologically, and how to tell if a drug is hitting its target. Now let's zoom out to the bigger picture again. How do you actually design a study that can prove these effects in potentially thousands of people over many years, keeping in mind all these complexities we've discussed? This sounds like a huge operational challenge. #6. Trial design considerations and innovations.
It absolutely is, and this is where the rubber really meets the road in translating geroscience. Designing human trials for geroprotective therapies requires this incredibly delicate balance. On one hand, you need rigorous scientific methodology to convincingly prove efficacy and safety. On the other hand, you need practical feasibility, given the long human lifespan and the inherent complexity of the aging process itself. It's a constant tension.
So what are the biggest challenges right off the bat, maybe in terms of who you even enroll in these studies?
Yeah, that's a critical starting point. First, population selection, power, and heterogeneity. To detect a meaningful change in age-related outcomes within a reasonable timeframe, trials often adopt a strategic approach. They tend to recruit older adults who are already at a somewhat higher risk of experiencing age-related events. The rationale is straightforward. Enrolling participants of advanced age, or maybe those with some pre-existing risk factors like minor chronic conditions or maybe just a slow walking speed, naturally leads to more outcome events like disease diagnoses or functional decline occurring within a shorter study duration. This significantly improves the trial's statistical power. The TAME trial, for instance, explicitly enrolls adults aged 65 to 80 who already have or are at high risk for an age-related disease or functional limitation, precisely to enrich the sample with potential events. However, this involves a trade-off. A very high-risk sample, say people in their late 70s with multiple serious conditions, might not be truly representative of the broader, healthier population of 50 or 60-year-olds who might eventually take a preventive aging drug long-term. So a common strategy is maybe to conduct phase 2 trials in a higher risk group to quickly establish a signal of efficacy, and then potentially confirm those findings in a broader, more general population in a larger phase 3 trial.
Right, that makes sense. And how do you deal with the huge variation? People age so differently.
That's the challenge of heterogeneity, and it's a major factor in any aging study. People age at widely different rates, and human populations are incredibly diverse biologically and environmentally. Therefore, trials must meticulously account for potential differences related to sex, ethnic, and genetic diversity, and the wide baseline health variability you see in any group of older adults. Including a diverse sample isn't just an ethical imperative anymore. It actually helps researchers detect if the intervention works consistently across all subgroups, or if its efficacy or safety might be modulated in certain profiles. Maybe it works better in men than women, or vice versa. This means actively implementing strategies like stratifying randomization, making sure you have roughly equal numbers of males and females, or balancing racial groups across the treatment arms and pre-specifying subgroup analyses in the trial plan to look for these potential differences. Then there's the challenge of sample size and event-driven design. As we've said, outcomes like mortality or the onset of dementia are relatively rare over the typical short periods of a clinical trial, especially in healthier populations. The consequence is that aging trials often require very large sample sizes. We're talking potentially thousands of participants who need to be followed for multiple years. This is a huge logistical and financial undertaking. To mitigate this, strategies include using those composite endpoints we discussed, which help increase the overall event rate. Another powerful strategy gaining traction is using an event-driven design. Instead of ending the trial at a fixed calendar date, say after 3 years, the trial protocol specifies that it continues until a certain predetermined number of outcome events, like 150 first occurrences of multimorbidity or death, have occurred across all participants. This significantly improves efficiency because the study concludes precisely when sufficient statistical information has been gathered to answer the primary question, regardless of the exact calendar time it took.
Okay, that's clever. But how on earth do you keep thousands of people taking a daily pill, maybe for years, especially when they don't even feel sick? Adherence. must be a massive headache.
It is a huge challenge, and that's our second major consideration: adherence and duration. In preventative trials, especially for aging where the participants are often healthy or only mildly impaired, adherence to the intervention over many years is both critically important and uniquely challenging. If participants don't perceive an immediate benefit, or if they experience even mild side effects, their motivation to take a daily pill might wane over time. This phenomenon is well known as adherence decay. So, trials have to employ active strategies to tackle this. An initial run-in period where everyone takes a placebo for a short while can help screen out individuals who struggle with compliance right from the start. And ongoing support throughout the trial is crucial. This might involve regular counseling, automated reminders via smartphone apps, periodic check-ins, maybe even using smart pill bottles, or sometimes measuring drug levels in the blood to objectively gauge compliance. It's also vital to collect detailed adherence data and actually plan for expected levels of non-adherence in the initial power calculations to ensure the trial is still robust. Handling dropout is also vital. Older participants might drop out for various reasons. Trials need pre-specified statistical methods to handle missing data and often involve geriatric specialists in designing participant support programs to minimize loss to follow up. Third, and this is where things get really innovative, we see a lot of movement towards platform and adaptive trial designs. There's growing interest, and rightly so, in platform trials as an incredibly efficient way to evaluate multiple potential therapies simultaneously for aging interventions. The concept is simple but powerful. One master protocol allows researchers to test several different treatments in parallel against a shared control group. Crucially, this design has the inherent flexibility to add new experimental arms as new promising candidates emerge, or drop ineffective arms mid-study based on interim data.
Wow, that sounds incredibly efficient compared to running separate trials for everything.
It is. It saves enormous amounts of time and resources because you're not setting up a whole new trial infrastructure and placebo group for every single drug candidate. It effectively gives each active arm a larger control cohort to compare against, boosting statistical power. And the adaptive features are key. Built-in interim analyses allow researchers to stop an arm early for futility if it's clearly not showing benefit. Even more sophisticated designs use response-adaptive randomization, where, based on early positive signals, more participants can be allocated to the more promising arms of the trial. This weeds out ineffective interventions quicker and, importantly, ethically assigns more participants to therapies that appear to be working, which is a major advantage in a long-term preventive trial setting. Platform trials are particularly well-suited for geroscience because we have numerous candidate interventions, metformin, rapamycin, various senolytics, AKG, etc., but limited time and funding to test each one sequentially in massive years-long trials. A platform approach allows for continuous evaluation and maximizes the learning gained per patient enrolled. And finally, complementing these traditional trials, we have the growing role of real-world evidence, RWE, and pragmatic trials. Beyond the highly controlled environment of classical randomized controlled trials, RCTs, the field of geroscience is actively exploring pragmatic trial designs and leveraging real-world evidence to gather additional efficacy and safety data in more realistic settings. Pragmatic trials are often conducted in routine clinical care settings. They have minimal exclusion criteria, making the population more representative, and they test interventions that can be easily embedded into real-life practice. They might compare a new intervention against standard care rather than a placebo, focusing on outcomes that matter directly in day-to-day life, like falls or hospitalizations. The LIFE study, for example, was somewhat pragmatic in testing physical activity versus health education in older adults to reduce mobility disability, showing that a simple walking program worked. Real-world evidence comes from analyzing large healthcare databases, patient registries, or even data from wearable devices. Observational studies using RWB, for instance, have shown that people with type 2 diabetes taking metformin seem to have lower all-cause mortality and incidence of some other diseases compared to non-users, providing valuable hypotheses. And a really powerful policy mechanism that bridges trials and real-world use is coverage with evidence development, CED. We touched on this earlier. It's where public insurers like Medicare could provisionally cover an aging intervention, but only on the condition that beneficiaries enroll in a data collecting program or trial. This effectively transforms real-world use into a pragmatic study, providing companies with a revenue stream while simultaneously gathering crucial real-world evidence to confirm the therapy's long-term value. It treats prevention as a public good, where society invests in learning.
This is a truly ambitious and innovative landscape for clinical trials. But it brings us back to a fundamental point. When you're talking about giving potentially healthy people a drug for years, maybe even decades, the stakes on safety are incredibly high. So how do they monitor for risks, specifically in this preventative context? #7. Safety monitoring unique to geroprotectors.
You've hit on perhaps the most critical aspect, the absolute cornerstone for developing geroprotective therapies. When interventions are designed to be given to generally older and often asymptomatic individuals for prevention, not treatment of an existing disease, safety is paramount. It takes precedence over almost everything else. Unlike treating a severe illness where some significant side effects might be tolerated in exchange for a life-saving benefit, these drugs are intended for long-term, potentially decades-long, use in people who are basically healthy or have only mild conditions. In this context, even seemingly modest risks could accumulate over that long duration and dramatically offset any potential long-term benefits and health span. Therefore, the safety bar is set exceptionally high.
Okay, so what are the specific areas of concern? What are researchers really watching out for?
There are several key safety areas that receive special attention in these trials. First, infection and immune effects. This is a big one because some aging interventions are designed to modulate or maybe even tamp down certain immune pathways associated with aging, like chronic inflammation. A prime example is M2 inhibitors like rapamycin, which are known to have mild immunosuppressant effects, particularly at higher doses used for transplant medicine. So trials must monitor infection rates very, very closely in participants taking these drugs compared to placebo. Interestingly, and somewhat counterintuitively, for low-dose everolimus, which is a rapamycin derivative or Rapalog used in older adults, an initial concern about increased infection susceptibility was observed. However, in one notable trial, low-dose M2 inhibition actually seemed to enhance immune function, improving the response to flu vaccines and, surprisingly, reducing the overall rate of infections reported by participants. This really highlights that dose matters. While low intermittent doses might be beneficial for immune health, higher or continuous doses could potentially increase infection risk. Therefore, the Data and Safety Monitoring Board, or DSMB, which is an independent committee overseeing the trial, always sets predefined stopping rules specifically related to infection rates. If serious infections climb significantly in the treatment arm, they can pause or halt the trial. Routine lab tests like white blood cell counts are monitored regularly. Second, a related area is wound healing and recovery. Certain drugs that slow down cellular processes like cell growth, and again, rapamycin is a known example here, can potentially impair wound healing. In surgical contexts, for instance, rapamycin is typically held before and after surgery to avoid poor healing outcomes. So in aging trials, researchers must be particularly mindful if participants require surgeries during the trial or sustain injuries like falls or cuts. Any signals suggesting delayed wound healing or problems with tissue repair, things like non-healing foot ulcers, or slow recovery from fractures in the group receiving the drug would raise significant safety concerns. Trials often collect specific data on falls and fractures and track how well participants recover from them, ensuring the drug doesn't inadvertently interfere with the body's essential repair mechanisms while trying to promote longevity. Third, there are potential metabolic side effects, things like effects on blood glucose levels, lipids, or even blood counts like anemia. Many potential aging drugs affect metabolism broadly because aging itself is deeply intertwined with metabolic regulation. Rapamycin, for instance, has been associated with slight insulin resistance or changes in lipid profiles in some contexts, although again, recent studies using low doses in healthy older adults did not show significant negative changes in glucose or lipids over a few months. Anemia is another potential concern for some interventions that might perhaps suppress bone marrow function slightly or alter iron metabolism. And a highly relevant example here is metformin. While generally considered very safe after decades of use, we know that chronic metformin use can lead to vitamin B12 deficiency in a significant fraction of patients. This deficiency, if unaddressed, can cause anemia and even neurological symptoms like neuropathy. This is a very pertinent example. Trials like TANE explicitly incorporate routine monitoring of B12 levels and have plans to treat any deficiencies that arise. It highlights how even subtle nutritional side effects need careful attention in long-term preventive drug use. Other standard metabolic markers like liver enzymes, kidney function, and electrolytes are, of course, routinely monitored as well.
Okay, that makes sense. And what about the big one, the really long-term concern? Cancer. Is there a risk there?
Fourth, cancer and long-term safety signals. This is a somewhat paradoxical concern, but an important one. While the ultimate goal of many geroprotectors is actually to reduce cancer incidence over the long run by targeting fundamental aging mechanisms that contribute to cancer development, modulating certain biological pathways could, in theory, potentially increase short-term tumor risk. For example, if a drug stimulates stem cell function, is there an unintended trade-off related to cancer? Therefore, trials must monitor cancer incidence very carefully, even if the early phase trials aren't statistically powered to detect small differences. An unexplained increase in any particular type of cancer, or even overall cancer rates in the treated group compared to placebo, would be an immediate red flag and likely stop development. Conversely, of course, if treated participants actually show fewer new cancers developing during the trial, that would be a hugely significant positive finding supporting the drug's benefit. It's worth noting that so far, short-term studies of rapalogs in seniors have not shown worrying cancer signals, and some preclinical and even clinical data suggest rapamycin might lower the risk of certain cancers. But continuous vigilance over the long term is absolutely paramount. And fifth, encompassing many of these, is monitoring for effects on general geriatric syndromes. Trials also proactively monitor for impacts on common geriatric syndromes that reflect overall health and function in older adults, things like falls, cognitive changes, and sarcopenia, which is age-related muscle loss. The fundamental principle is that the intervention shouldn't worsen any aspect of overall health span while trying to improve another. For example, if a drug dramatically improved cardiac health but inadvertently led to increased muscle loss or accelerated cognitive decline, that would be a major problem for its overall benefit-risk assessment. So functional measures that might be efficacy endpoints like gait speed, grip strength, chair stands, cognitive tests also serve double duty as crucial safety endpoints, ensuring no unintended acceleration of decline in any important domain of health.
Right. So it's a constant vigilant balancing act, monitoring a wide range of potential issues specific to intervening in the aging process itself.
Absolutely. And rigorous oversight is key. As mentioned, every geroscience trial is overseen by that Independent Data and Safety Monitoring Board, DSMB, which regularly reviews the unblinded safety data. They have the authority to recommend pausing or stopping the trial based on predefined stopping rules if specific adverse events exceed a certain threshold. Importantly, safety monitoring is meticulously tailored to the specific mechanism of the anti-aging intervention being tested. For example, for senolytics, which target senescent cells, trials might pay close attention to platelet counts or liver enzymes, as some early candidates had chemotherapy-like effects. For NAD boosters, maybe monitoring blood pressure is important. The good news, as we said, is that many leading candidates are repurposed drugs like metformin or rapamycin, for which we already have extensive safety profiles from their decades of use in other conditions. The main task now is to confirm that using them in healthier, older adults for long-term prevention doesn't reveal new or different safety issues specific to this population or duration of use. But the bottom line is that the benefit-risk calculus for prevention is exceptionally stringent. Tolerating even moderate side effects is generally not acceptable if the person has no existing illness. The intervention must be proven to be extremely safe given its intended long-term preventive nature for a potentially broad, healthy population. Regulators will undoubtedly demand a very large safety database, accumulating many thousands of patient years of exposure before approving a drug for a broad indication like delaying aging.
Okay, that's crystal clear. Safety is job number one. So even if the science is sound, the trials are well designed for efficacy and safety, there's still that really practical question of how you actually produce enough of these therapies, especially the more cutting-edge ones. It's not just about making a simple pill anymore, is it? #8, manufacturing and CMC hurdles, cell gene therapies.
You're absolutely right. This is a part of drug development that often goes completely unnoticed by the public, but it's often a make or break factor for actually bringing new therapies to the world. Can we actually make enough of it consistently, affordably, and safely? And for longevity therapies, especially the more advanced ones, this represents a whole new level of challenge. While scientific breakthroughs grab headlines, the practicalities of manufacturing often determine whether a therapy can ever become widely accessible. Not all potential longevity therapies are simple pills or small molecules that can be synthesized cheaply at scale. Many cutting-edge ideas involve incredibly complex biological entities. Things like gene therapies, where you're introducing new genetic material, stem cell therapies involving living cells, perhaps CAR therapies engineered to target senescent cells, and other advanced biotechnologies. These advanced therapies bring substantial manufacturing and CMC, that's chemistry, manufacturing, and controls hurdles that can significantly slow down or even completely halt the translation from promising lab studies to human application, regardless of how well they work biologically in principle. The core question becomes, even if a gene therapy works beautifully in a petri dish or a mouse to rejuvenate tissues, can it be feasibly produced for thousands or even millions of patients at a reasonable cost and with consistent quality?
Okay, so what's so uniquely difficult about making these new types of treatments compared to, say, a traditional pill?
Well, let's dive into the specific cell and gene therapy manufacturing challenges. First, there are huge scalability issues. Right now, many cell therapies, for example, infusions of stem cells from a donor, allogeneic, or maybe CAR T cells engineered to kill senescent cells are almost handcrafted. It's artisanal almost. Cells, whether they're harvested from the patient themselves, autologous therapies, or come from a universal donor bank, allogeneic therapies, need to be expanded, sometimes genetically modified and processed in highly specialized, often small-scale, clean room facilities. This entire process is inherently time-consuming, incredibly labor-intensive, and extremely costly. A major bottleneck for scaling up production is the lack of fully automated, high-throughput bioprocessing tools and systems. Manufacturing is often done in small batches with many manual steps, which not only drives up the cost per dose astronomically, but also introduces variability between batches, making consistency a real challenge. For autologous therapies, where each patient's own cells are the starting material, you have the N of 1 manufacturing challenge. This means each patient's product is essentially unique. This makes standardization and achieving economies of scale extremely difficult. You're essentially setting up a bespoke manufacturing process for every single patient. This difficulty is partly why the industry is increasingly shifting towards allogeneic approaches for potential aging treatments, using universal donor cells or off-the-shelf engineered cell lines. These could theoretically be produced in larger standardized lots and stored, potentially drastically reducing the cost per dose and making widespread use more feasible. However, allogeneic therapies sometimes provoke an immune rejection response in the recipient or might be less efficacious than autologous cells, so this is still an active area of intense innovation. For example, using gene editing techniques like CRISPR to make donor cells invisible to the immune system, hypoimmunogenic. Then, for gene therapy, viral vector manufacturing, the challenge is different but equally daunting. Manufacturing the extremely high doses of viral vectors like AAV or lentivirus needed for potential widespread systemic use to target aging across the body is a massive undertaking. Key challenges include achieving sufficient yield, how much active, functional vector can actually be produced per manufacturing batch, and ensuring a high purity, effectively removing contaminants, empty viral shells, capsids, and other impurities from the final product. Producing these viral vectors at the scales potentially needed for a common condition like aging currently strains global manufacturing capacity significantly. Furthermore, the cost of goods, or COGS, for producing a single gene therapy dose can currently run into the hundreds of thousands or even millions of dollars. This is completely untenable for a preventative treatment intended for a broad, generally healthy population. Huge efforts are underway across the industry to develop better bioreactors, more efficient producer cell lines, and advanced purification methods to significantly increase viral vector yield and drastically reduce the cost per dose.
Wow. And I imagine regulators need absolute certainty that everything is exactly right batch after batch.
Absolutely. Regulators like the FDA impose extremely rigorous regulatory demands for CMC data. They require extensive CMC data as a foundational part of any drug application for these complex biologics. This includes demonstrating unwavering consistency between batches, ensuring every manufactured lot is virtually identical in composition and function, proving the stability of the product over time under defined storage conditions, ensuring the complete absence of contaminants like bacteria, fungi, or residual chemicals, and, critically for viral vector therapies, proving the absence of any replication-competent viruses that could potentially cause infection. Many longevity biotech companies working on these advanced modalities are effectively platform companies. They are simultaneously building their novel manufacturing processes and technologies alongside the development of the therapeutic product itself. It's a dual challenge, and the scaling up problem is very real. If an aging gene therapy shows fantastic promise in a small phase 1 trial, maybe in just 20 people, scaling up production for a large phase 3 trial involving, say, 2,000 people means producing orders of magnitude more vector. This transition from small-scale to large-scale manufacturing is often the point where many promising programs falter because their initial, perhaps academic-level production method simply doesn't scale linearly or cost-effectively. Related to this are the quality control QC hurdles. With standard small molecule pills, routine analytic chemistry can characterize the product precisely. But with living cells or complex viruses, characterization is far more complex and less standardized. It requires a whole suite of sophisticated biological assays to ensure things like gene expression profiles are correct, cells have the right surface markers, or functional assays confirm the intended biological activity. For example, release testing for a batch of cell therapy might involve time-consuming biological assays to confirm its potency. For example, do the senolytic CAR-Pro T cells actually kill senescent cells effectively in a lab dish test before they can be released for patient use? So companies are investing heavily in manufacturing innovations. We're seeing the development of fully closed, automated cell culture systems designed to minimize manual labor, reduce contamination risk, and improve consistency batch to batch. Improved bioprocessing techniques, like using microfluidic devices for cell processing or AI to optimize culture conditions, are aiming to break the current capacity ceiling that limits many cell therapy manufacturers from meeting potential larger demand. And the field is actively investigating ways to manufacture biologics cheaper more broadly. For instance, using mRNA therapies, like the COVID vaccines, is being explored as a potentially more scalable and cost-effective alternative to viral gene therapies for delivering certain longevity factors, since mRNA can be produced synthetically more readily.
It's sobering. A brilliant lab discovery, a potential breakthrough in aging biology, could fail simply because it can't be manufactured reliably and affordably at the scale needed.
Precisely. A therapy intended for potentially millions of generally healthy people sets an extremely high bar for manufacturing. It needs to be cost-effective, consistently safe, free of any adventitious agents, reproducibly manufactured, and highly scalable. The situation is quite analogous to vaccine production, where massive scale is needed to reach healthy populations worldwide, and the COVID-19 pandemic starkly illustrated how manufacturing capacity and logistics can become the primary bottleneck limiting rollout, even when the science is sound. Longevity therapeutics, especially advanced ones, will likely require a similar global infrastructural commitment. Therefore, addressing these manufacturing and CMC challenges is not an afterthought. It's an integral part of the entire process of bringing gerotherapeutics from the lab bench to the patient bedside successfully. Scientists developing these therapies must collaborate closely with bioengineers, manufacturing experts, and regulatory specialists from the very earliest stages of development to ensure their exciting discoveries don't languish due to an inability to actually produce them at scale.
This makes so much sense. It really highlights that it's not just about the underlying science or even the clinical trials, but the entire ecosystem needed to develop, manufacture, and deliver these therapies. And that brings us to perhaps the biggest question of all. If we do prove these therapies work, who pays for them? And how do we ensure they're actually accessible to everyone who could benefit, not just the privileged few?
These are absolutely profound questions, and they really get to the heart of the complex economic and policy landscape surrounding preventive medicine, especially when applied to something as fundamental as aging. Even if a therapy is scientifically proven beyond doubt to extend health span, the question of who will pay for it and how remains a significant, perhaps even existential, challenge for the field. The core problem lies in what economists often refer to as the public good aspect of preventive medicine. The benefits of successfully preventing age-related decline, things like avoided disability, extended years of healthy, productive life, reduced burden on caregivers, these benefits accrue broadly to individuals, families, and society as a whole. However, it's inherently difficult for private companies operating in a market economy to capture sufficient profit directly from something that doesn't happen, like a stroke that was prevented 5 years down the road or a decade of expensive nursing home care that was averted entirely. Developing these therapies involves extraordinarily high upfront research and development costs and long timelines, while the private financial returns for prevention can be uncertain and delayed compared to treatments for acute, already present diseases where the value proposition is more immediate. Therefore, intervening in aging is, in many ways, fundamentally a public good, offering widespread societal benefits that extend far beyond direct commercial profit potential for any single entity.
Okay, so if it's a public good that's hard for the private market to fully monetize, how do you incentivize companies to actually invest the huge amounts of money needed to develop these therapies?
That's the crux of the policy challenge. We need to consciously create incentives to spur investment in this area. One concept that's often discussed is potentially extending exclusive market rights for truly proven geroprotectors. This would be somewhat analogous to the incentives provided under orphan drug legislation, which encourages companies to develop treatments for rare diseases that might not otherwise attract sufficient investment due to small market size. Granting a period of market exclusivity could help companies recoup their substantial R&D costs for aging therapies. Another potentially powerful policy approach we've touched upon is coverage with evidence development, or CED, implemented by payers. Remember, under CED, a major payer like Medicare or perhaps a large private insurer agrees to provisionally cover a newly approved intervention on the condition that the patients who receive it are enrolled in a study or a registry designed to continuously collect real-world outcomes data.
Right, so companies get paid earlier, which is a huge incentive, and society gets more data to confirm the value. It sounds like a win-win, or at least a way to share the risk.
Exactly. It offers a dual benefit. It gives pharmaceutical companies a vital revenue stream much earlier than they might otherwise get it, especially while long-term outcome evidence is still emerging. This dramatically changes their financial modeling. Simultaneously, it allows society, through the payer, to gather crucial real-world evidence to confirm the therapy's long-term value, cost-effectiveness, and safety in a diverse real-world population. It essentially shares the risk between the developer, the payer, and society. If the drug truly prevents expensive downstream outcomes like hospitalizations or long-term care, then everyone benefits. The payer saves money in the long run, patients live healthier lives, and the company has a viable market. If the long-term benefits don't materialize as hoped in the real world, coverage might be adjusted or withdrawn based on that evidence. CED isn't just theoretical. It has been successfully used for certain new medical technologies in the past, like those beta amyloid PE scans for Alzheimer's diagnosis. For an aging drug, a CED approach could allow earlier patient access while simultaneously mandating ongoing outcomes tracking, perhaps through claims data and periodic functional assessments, thus incentivizing both patients and companies to participate actively in generating the necessary long-term evidence. Then there's the absolutely critical issue of cost-effectiveness and equitable access. Policymakers and health economists are increasingly engaging with the idea that preventative drugs for aging, if priced reasonably, could actually be cost-saving for healthcare systems in the long run. This is because they could potentially avert massive downstream expenditures associated with managing chronic diseases, hospitalizations, and long-term care like nursing homes. However, there's a huge potential pitfall. If such a breakthrough drug is launched with a prohibitively high price tag, like some specialized gene therapies today costing hundreds of thousands or even millions per treatment, then access would inevitably be severely limited. This could dramatically worsen existing health inequalities, creating a scenario where only the wealthy can afford to buy significantly more healthy years. This raises a profound ethical imperative. Ethicists and public health experts argue strongly that ensuring equitable access to bona fide anti-aging therapies, should they emerge, is crucial to avoid deepening social divides. This might require governments to play an active role, perhaps in negotiating prices, providing subsidies, or implementing other mechanisms to ensure affordability, recognizing the inherent public good nature of extending population health span. Some have even proposed innovative financing models like prize funds, large rewards for achieving predefined health span goals, or advanced market commitments, government guarantees to purchase a certain volume if successful, which could decouple the R&D incentive from high per-unit pricing and better align incentives with broad population health benefits.
And the stance of regulators themselves plays a big part in shaping this whole landscape, doesn't it? If they start recognizing aging differently.
It absolutely does. From a regulatory recognition perspective, if aging itself, or perhaps a well-defined aspect of it like multimorbidity or frailty, were eventually formally recognized by agencies like the FDA as a treatable condition or a valid indication for drug approval, perhaps spurred by the success of a trial like TAME, it would fundamentally open new pathways for drug labeling and reimbursement. The TAME trial, as we've discussed repeatedly, is seen by many in the field as potentially proving to medical agencies that aging or its consequences can be a therapeutic indication. Success here could create conditions for defining new approval types and provide a powerful incentive for pharmaceutical companies to invest much more heavily and confidently in this field. It could be the first domino that validates the entire approach and attracts significant industry investment that is currently hesitant due to regulatory uncertainty. And finally, underpinning all of this, coverage policies by insurers and national health systems will significantly influence the uptake and ultimate impact of any successful aging intervention. If an approved aging intervention is classified and covered as a preventive service, similar to how vaccines or certain cancer screenings are treated, then insurers may cover it widely, possibly even with minimal or no copay for patients, as mandated by the Affordable Care Act in the U.S. for recommended preventative services. Conversely, if payers view it skeptically, perhaps classifying it as a lifestyle drug or deeming it not medically necessary according to traditional definitions, then coverage could be widely denied. This would severely limit its use to only those individuals who can afford to pay entirely out of pocket, reinforcing those equity concerns. Therefore, a clear demonstration through rigorous trials and health economic analyses of reduced disease burden and tangible healthcare cost savings will be absolutely crucial to convince payers of the value proposition. Health economic studies are already underway, even as the clinical trials run, attempting to model the potential long-term healthcare cost savings if a drug successfully delays nursing home entry by a few years or halves the incidence of Alzheimer's disease. These models aim to strengthen the case that even an initially expensive drug can yield significant net financial benefits for the healthcare system over the long term by replacing years of very high healthcare expenditures with healthier, less costly life years.
It's clear that aligning the financial incentives and policy frameworks with the scientific goal of extending healthy life is just as critical, maybe even more complex, than the scientific breakthroughs themselves.
Absolutely. Prevention, historically, has been a hard sell in healthcare systems that are often structured primarily to reward acute treatments for existing diseases. However, with the demographic reality of the looming silver tsunami, the rapid aging of populations in many countries worldwide, the economic calculus is undeniably changing. Governments and insurers are increasingly recognizing that investing proactively in health span and keeping people healthier for longer could yield enormous dividends, not just in improved quality of life, but also in reduced long-term healthcare costs and sustained economic productivity. So in the future, we might see the emergence of more innovative policies specifically designed to foster this field, perhaps things like tax credits for preventive drug development, more public-private partnerships for large-scale trials, similar perhaps to pandemic preparedness initiatives, or even international collaborations to pool resources for evaluating and sharing data on promising aging interventions. And that regulatory evolution, formally recognizing aging itself, or key aspects of it, as a modifiable risk factor within regulatory and reimbursement frameworks will be absolutely critical to unlocking the field's full potential. It's certainly a hopeful sign that aging biology is now such a prominent topic, with significant government initiatives like the NIH's Geroscience Interest Group and even international bodies like the WHO considering adding healthy lifespan metrics to their global health targets. The groundwork is slowly but surely being laid to treat aging not as an inevitability to be passively accepted, but as something we can and should actively seek to modify for the public good, benefiting society as a whole, not just individuals.
This is clearly a future-facing vision, requiring unprecedented collaboration across science, medicine, economics, and policy. So let's try and bring all these incredibly complex pieces together. How might they converge in a concrete clinical trial? Let's walk through an example to see how a real Phase 2b geroprotector trial might actually be designed today using all these principles we've discussed.
Excellent idea. Let's try to synthesize everything into a hypothetical but realistic example. Imagine we have a promising new candidate geroprotective drug, let's call it RejuveX. We want to design a robust Phase 2b clinical trial to get a really good sense of whether RejuveX actually impacts the aging process and improves health span in humans in a way that incorporates many of the best practices and innovative concepts we've explored. This trial would be designed to provide strong evidence to decide whether to proceed to a much larger, definitive Phase 3 study.
Okay, so starting with the basics, who'd you enroll in this hypothetical RejuveX trial?
We'd probably aim for a reasonably large Phase 2b size, maybe around 800 adults. We'd likely target an age range like 70 to 79 years old. This age group is old enough to have a sufficiently high rate of age-related events occurring over a few years to give us statistical power, but hopefully not so frail that intervention is too late or confounded by too many existing severe diseases. The key would be selecting participants who have some evidence of aging risk but are not yet experiencing advanced end-stage disease. We need that balance between event rate and generalizability. So the inclusion criteria might specify that participants must meet either of the following. 1, they have at least one existing but well-controlled age-related chronic condition, think maybe stable hypertension or osteopenia or mild stable heart disease. This ensures they have some room to benefit from a preventive intervention. Or alternatively, they show evidence of being in the fastest, say, 20% of biological agers for their chronological age, as assessed by a validated biomarker panel before enrollment. We could, for instance, use a specific cutoff score on the Dunedin-Pace epigenetic clock to identify these faster agers. This leverages those cutting-edge biomarkers right up front for patient selection, a form of precision medicine. And crucially, to account for that heterogeneity we discussed, randomization would be meticulously stratified by key factors like sex, aiming for roughly 50-50 balance, and maybe baseline frailty status, categorizing participants as robust versus pre-frail or mildly frail using a standard assessment to ensure these groups are balanced across treatment arms and allow for potential subgroup analysis later.
Okay, population selected. How would the drug actually be tested in this group?
Participants would be randomized 1.1, meaning equal chance to receive either the active drug, Rejuvex, or an identical-looking placebo taken orally once daily. The trial would absolutely have to be double-blind, meaning neither the participants nor the researchers, clinicians, or study staff interacting with them know who is receiving the active drug versus the placebo. This is fundamental to minimizing bias in reporting symptoms, assessing outcomes, everything. And we'd likely incorporate that 1-month placebo run-in period before the actual randomization. Everyone gets placebo for a month. This is invaluable for screening out individuals who might struggle with taking a pill every day or who experience unrelated health issues early on that might complicate their participation in the main long-term study. It helps ensure you start the core trial with a more adherent and stable group.
Right. And what would be the main finish line, the primary measure of success for Rejuvex?
The primary endpoint would almost certainly be a composite time-to-event endpoint. This is really emerging as the standard for capturing the broad, multi-systemic benefits expected from a true geroprotector. We would pre-specify the definition very carefully. It would be the time until a participant experiences any one of the following pre-specified events, whichever occurs first. Perhaps a new diagnosis of a major chronic disease from a defined list, AG, coronary heart disease event, stroke, invasive cancer, or moderate dementia. Or maybe persistent major mobility disability, rigorously defined, for instance, as being unable to walk 400 meters on 2 consecutive semi-annual visits, ensuring the disability is sustained and clinically meaningful. Or simply death from any cause. We would plan to follow participants for a defined period, say up to 3 years, and importantly, the trial would likely be designed using that event-driven approach we talked about. We wouldn't just stop at 3 years regardless. We'd target a specific total number of primary composite events occurring across both groups, maybe 150 events, for example. The trial continues until that number is reached, which optimizes efficiency and ensures adequate statistical power. The underlying hypothesis is that if Rejuvex truly slows aging broadly, we'd expect to see significantly fewer people hitting one of these adverse outcomes in the treatment arm over the 3-year period compared to the placebo group.
Okay, that makes sense for the main goal. But what about those all-important biomarkers and other measures? What secondary outcomes would you track?
Absolutely essential. We'd have several key secondary endpoints to provide a more complete picture. First, physical function. We'd measure changes in objective physical function over, say, 2 years, specifically assessing things like gait speed, walking speed in meters per second, and grip strength. We'd also analyze the proportion of participants experiencing a clinically meaningful decline in gait speed over time, which captures something very relevant to patients' daily lives. Second, frailty. We track the change in a validated frailty index, perhaps one based on the accumulation of around 30 predefined health deficits, comparing the change from baseline to 2 years between the groups. This gives us a continuous measure of overall health status and vulnerability. Third, and crucially for mechanism, biological age markers. The trial would absolutely measure the Dunedin-Pace epigenetic aging rate at baseline, 1 year, and 2 years. This directly assesses if Rejuvex is managing to slow the fundamental pace of biological aging at the molecular level. We'd likely include other related markers too, like maybe the Grimmage epigenetic clock acceleration score, and perhaps a panel of key blood inflammation markers like IL-6 and CRP. These secondary outcomes are vital for interpreting the primary result. For instance, if the primary composite endpoint result is statistically borderline or neutral, but Rejuvex significantly improved these biological aging markers and functional measures, it would provide strong supportive evidence and potentially justify moving forward with further research, maybe a larger or longer trial, or targeting a specific subgroup. Conversely, if the primary endpoint is positive, seeing positive changes in the biomarkers would strongly bolster the biological plausibility and our confidence that the drug is truly acting on aging pathways.
Right, they add crucial context. And given this is likely a long-term preventive drug for older adults, safety must be monitored incredibly carefully. How would that be structured?
Safety monitoring would be absolutely comprehensive and multi-layered. The primary safety endpoints would include tracking all serious adverse events, SAEs, such as hospitalizations for any reason, life-threatening events, or deaths. We'd pay special attention to predefined categories of interest based on the drug's expected mechanism or findings from earlier studies, perhaps focusing particularly on infections, hematological changes, like changes in white blood cell counts or hemoglobin, and metabolic changes, glucose, lipids, liver function, kidney function. Our independent DSMB would have very clearly defined stopping rules for safety. For example, the protocol might state that if the combined rate of serious infections requiring 4V antibiotics or hospitalization significantly exceeds that of the placebo group by a predetermined statistical margin, or if an unexpected pattern of unexplained excess mortality appears in the treatment arm, the DSMB has the authority and obligation to recommend pausing or stopping the trial immediately. Lab monitoring would be frequent, especially early on. Maybe a monthly complete blood count, CBC, and comprehensive metabolic panel for the first 3 months to catch any acute effects. Then the frequency might reduce to, say, quarterly thereafter. We'd pay specific attention to any concerns raised in preclinical studies, for instance. If animal studies hinted at mild myelosuppression, bone marrow suppression, we'd watch white cell and hemoglobin levels very closely. And based on the metformin example, we'd likely include nutritional monitoring, perhaps checking vitamin B12 and folate levels annually, anticipating potential long-term effects on nutrient absorption that might need management. The DSMB would formally review all the unblinded safety data at regular intervals, perhaps twice per year, to ensure ongoing participant safety throughout the trial.
And for these multi-year trials, adherence is always a major factor that can undermine the results. How would you try and ensure participants actually stick with taking the Rejuvex or placebo every day?
Adherence is indeed critical, especially over a long duration. The plan would need multiple components. We might use technology-assisted support. Participants could receive adherence support via a simple smartphone app that provides daily medication reminders and perhaps asks a quick weekly question about whether they've had any issues or forgotten any doses. We'd also want some objective measurement. Drug plasma levels could be sampled in a subset of participants at specific time points, maybe at 6 months, to objectively gauge average adherence and see if people are actually taking the drug. This provides concrete data beyond self-report. We have a plan for intervention for low adherence. Those participants found to have very low drug levels might receive a gentle, non-judgmental intervention, maybe brief counseling from study staff on the importance of adherence or troubleshooting practical barriers they might be facing. We have to anticipate adherence decay over the long duration. So the trial might employ practical strategies like providing medication in pre-sorted daily blister packs or pill packs, and potentially involving family members or caregivers, with participant consent, of course, where appropriate to help participants remember their daily dose. Maintaining motivation is also key. At each study visit, staff would likely reinforce the overall health goals of the study and the importance of each participant's contribution, helping to motivate continued adherence. And finally, in the analysis phase, while the primary analysis would be by intention to treat, including all randomized participants regardless of how well they adhered, the study would also pre-specify an adherence-adjusted analysis. This might involve using the measured blood drug levels as a variable in the statistical model, or perhaps performing a per-protocol analysis that looks only at participants who took, say, at least 70% or 80% of their assigned doses to better understand the drug's potential effect under ideal compliance conditions. The trial design would also likely include one planned interim analysis for efficacy and futility, maybe occurring around 18 months or after about half the target number of events, say 75 events, have occurred, whichever comes first. This analysis would be governed by strict statistical rules, often using something like O'Brien-Fleming boundaries, to control the overall risk of making a false positive claim, type I error, while still allowing the trial to stop early under specific circumstances. If the primary endpoint difference at the interim look is overwhelmingly positive, indicating clear and convincing efficacy, the trial could potentially be stopped early. This is ethically advantageous if the benefit is clear, allowing participants in the placebo group to potentially access the effective treatment sooner. Conversely, if the interim analysis shows there's a very low probability based on the data accumulated so far of the trial ever demonstrating a statistically significant and clinically meaningful difference by its planned end, futility, the trial might also be stopped early to conserve resources and spare remaining participants from continuing on an apparently ineffective treatment. For enrollment and follow-up, because this is a long-term prevention trial requiring sustained engagement, the design would incorporate significant participant engagement strategies. This might include things like quarterly newsletters with general health tips, carefully crafted not to reveal group assignment, and perhaps providing mild, standardized exercise and diet recommendations to all participants as part of standard supportive care. This ensures both groups benefit from general health advice and can act as a kind of active control aspect. And a really forward-looking approach would be to leverage real-world data, RWD, to supplement the trial data. Participants would provide consent at the start of the trial to allow researchers to track their electronic health records, EHRs, and potentially their Medicare or other insurance claims data over the course of the study and perhaps even beyond. This is incredibly valuable because if a participant misses a scheduled study visit, the researchers might still be able to capture crucial outcome information like whether they had a hospitalization for a heart attack or received a new cancer diagnosis through these linked real-world data sources. This significantly improves the completeness of the outcome data and enhances the pragmatic validity or real-world relevance of the trial's results.
Wow. Okay, this hypothetical Phase 2b trial for ReGVX really does seem to integrate all those complex layers we've discussed, from careful population selection and composite endpoints to biomarkers, safety, adherence, and even leveraging real-world data.
Exactly. The goal of such a design would be to provide a truly robust and multifaceted test of ReGVX's potential impact on the human aging process and overall health span. And the next steps would depend heavily on the results. If the trial successfully hits its primary endpoint with a clinically meaningful effect size, say, it shows a hazard ratio of 0.70 for the composite endpoint, meaning a 30% reduction in risk for those on ReGVX. And this is supported by positive trends or significant improvements in key secondary measures like slower biological aging markers and better physical function. Then it would provide compelling evidence to greenlight a much larger, longer, and likely more expensive Phase 3 registration trial, potentially enrolling an even broader population to confirm the findings. On the other hand, if the trial perhaps fails to meet statistical significance on the primary endpoint, but nonetheless shows strong and consistent positive changes in the biological aging biomarkers and maybe some functional measures, the developers wouldn't necessarily just give up. They might use that information to refine their approach, perhaps considering a higher dose, exploring a combination therapy, or designing a future trial targeting a more specific subgroup who seem to respond best based on the biomarker data. The key is that by integrating multifaceted endpoints, robust biomarker panels, careful safety nets, adaptive trial elements, and real-world data linkage, such a trial design aims to maximize the learning from every single participant and every dollar invested. And that's precisely what's needed in this still relatively nascent but incredibly exciting and rapidly advancing field of human anti-aging therapeutics. #outro.
What an incredible journey through the complexities, the challenges, and the cutting-edge innovations in aging research. We've really unpacked everything from the biology in tiny mice right up to the global economy and policy.
It truly has been a deep dive. And I think it makes it abundantly clear that the transition of aging research from those often spectacular successes in mouse studies to therapies that are genuinely effective and safe in humans demands a fundamental rethinking of nearly every single aspect of clinical science and drug development. It's not a simple translation. I think our key takeaways from this whole exploration are pretty clear now. First, there's the absolute necessity of satisfying regulators by choosing appropriate indications and clinical endpoints that directly measure tangible patient benefits, that crucial imperative to show improvement in how patients feel, function, or survive. Second, the smart, strategic, and supportive use of biomarkers not as a standalone endpoints for approval yet, but as invaluable tools to provide early feedback on biological mechanisms, predict longer-term outcomes, and enrich our understanding. Third, the absolutely crucial role of innovation in trial design thinking creatively through adaptive trials, platform trials, and leveraging real-world evidence approaches is essential to efficiently test multiple therapies and overcome the inherent challenges posed by the long human lifespan. Fourth, the paramount importance of ever-cautious, vigilant safety monitoring, especially in this preventative context where interventions might be given to generally healthy individuals for many, many years, demanding an exceptionally stringent benefit-risk calculation. Fifth, that often overlooked but absolutely critical role of manufacturing scale, consistency, and quality, particularly for the advanced cell and gene therapies that represent some of the most exciting frontiers. And finally, the undeniable need for bold policy and economic incentives, things like CED, maybe new exclusivity models to catalyze prevention as a genuine public good, and critically ensure equitable access if and when these therapies become a reality. And while the current regulatory reality is that aging itself isn't formally recognized as a disease, it does feel like that landscape is gradually, albeit slowly, shifting. This is largely thanks to the hard work being done to develop and validate consensus endpoints like multimorbidity and frailty, and the progress being made by landmark trials like TAMI that are pushing the boundaries. The blueprint, I think, for avoiding those beautiful failures of the past and achieving meaningful success in human longevity is clearly emerging. This path, while it's undeniably complex, it's demanding, it holds the profound promise that what works biologically in the mouse world can, with ingenuity and rigor, indeed translate to tangible, life-enhancing benefits in the human world.
So what does this all mean for you? Listen again. It means that the ancient dream, really of adding not just more years to life, but adding more life to years, more healthy, functional time, is genuinely moving closer to reality. It's being guided now by incredibly rigorous science and truly innovative thinking across multiple fields. And as we learn more about the fundamental science of aging and about the complex systems needed to translate that science into real-world therapies, we realize we're not just passively waiting for the future of longevity. We're actively shaping it right now through these efforts. And this raises a really important, perhaps even philosophical question for you to consider. If a proven safe therapy emerged tomorrow that could significantly extend your healthy lifespan by, say, 5 or 10 years, what would you actually do with those additional healthy years? How would our society fundamentally change if a significant portion of its population experienced a dramatically compressed period of morbidity and disability at the end of life, truly adding vibrant life to those extra years? The deep dive continues for you. Thank you for joining us.